Hello, everyone. I'm really happy to be here, and I must say, I forgot how nerve-wracking it is to speak in front of actual people, so it's going to take me a while to lose the shaking in my voice. So I hope you don't mind that. So I'm going to talk about uncovering the spatial-temporal dynamics of language production. And first, before I do that, I'm going to talk to you a little bit about myself. As a primer for the gender bias discussion that we will have on Thursday. So I'm a French-American neuroscientist. I was born in Paris, and I moved to Rennes to start studying ethology. So that's the study of animal behavior, and I was actually really interested. The dream behind it was trying to understand or to know whether or not my cat actually was really understanding what I was saying to him. So, which actually I don't think is the case, but I found that out. So, but I was in fact really interested in what happens in the brain when we're speaking, right? How is it that we communicate with one another? So, in order to do that, I moved to Marseille and I started working with Xavier Alario, who is right there and who left, but he'll be back. Right there he is. Okay, and Boris Buhl, and started studying language productions. Right? So how is it that we use our brain to produce language? And that's where I got my PhD. And then I moved to California, so I took a long flight all the way to San Francisco, and while I was on that flight, I had an in-prep publication coming, right? So here was my daughter coming. So I had my daughter shortly after I arrived in Berkeley, and then just before I started my position in San Diego, I had another baby, and then I moved to San Diego, and that's where I am now. So anyway, just giving a little bit of context as to the other part of me, right? So I'm not just going to, just so we don't only talk about the content, right? And we think about the other parts of our lives, and hopefully that gives you a little bit to think about before Thursday, and hopefully helps you come out of your scientist shell as well. Okay. Thank you. All right, so we're moving into the science part. So there are many stages that are needed to produce speech. And that's when my voice is going to stop shaking, see? So this is a model by Leveld and colleagues. And so this is one of the famous models in language production. And so it basically shows you all the different things you need to do, or in fact, what your brain needs to do in order for you to get that sound out, right? So you need to find the concepts. You need to then find the word representations, which are also called lemmas. You need to find the morphemes, you need to find the phonemes, etc. And then finally, you're going to start speaking. And actually, we do this naturally 2 to 3 times per second, right, when we speak, which I'm doing right now. So we do this extremely fast, right? And there have been many studies trying to understand which brain regions are involved in helping us produce language. This is a meta-analysis by Kathy Price looking at PET and fMRI studies and showing all of the brain regions that we need, right, when we're speaking. So we're a bit past Broca and Wernicke, if you remember that from school. So we need, of course, much more of our brain to produce language. But as I said, we do a lot of things in very little time when we speak. So temporal resolution is actually needed to entangle speech production processes in real time. So I think using brain imaging methods that have a high temporal resolution is actually really important to understand speaking. However, speaking requires facial movements, right? And that's actually a major challenge when we want to use EEG and MEG to study speech production, right? So the main techniques with high temporal resolution that are available are EEG and MEG. And EEG is the most readily available, it's the cheapest, but EEG signal is also heavily polluted by electromyographic artifacts. So this is just showing you all of the, and some missing, but a lot of the muscles that are involved in, that are on our faces, and a lot of these muscles you need to produce speech. So in blue here, what you have is a EEG signal recorded while I was saying the word tractor, which you see the waveform of right underneath, right? So this is me saying tractor, actually in French, tracteur, and you can see the stop in the middle, right? So that you can nicely see the two syllables there. And what you see in blue, so the EEG signal, what you can see there is that there is a lot of EMG activity that you will actually record with the EEG signal, right? So it's the EEG activity, the EMG activity heavily pollutes the EEG signal, and that's a major challenge when you want to use this technique to study language production. All right. Okay. So this is what I'm going to talk about. So I'm going to be talking about how do we overcome articulation-related EMG artifacts. And then I'm going to be talking about a couple studies of EEG, using scalp EEG to study speech production, and then a little bit about intracranial EEG of speech production. And I'm going to start with how do we deal with these EMG artifacts. Okay, so a solution that we came up with when I was doing my PhD here in Marseille, I know we're in Aix, but it's right by, so is blind source separation based on canonical correlation analysis. Shorter acronym for that is BSSCCA. Okay, so how does this work? The idea is that EEG is more highly autocorrelated than EMG, right? So EEG theoretically is made up of a sum of oscillations, right? And so it's more autocorrelated in time. It tends to repeat itself more than EMG, which resembles white noise more. So what this canonical correlation analysis, or blind source separation based on canonical correlation analysis, is going to do is that it's going to separate sources, mathematical sources, based on their degree of autocorrelation. And we are going to remove the sources that are the least autocorrelated. So how do we do this? So first we process our raw data, so we high pass filter it. We remove eye blinks, and so in this study, we use the Gratton et al. Method, which is a simple regression method. And then we segmented the data around stimulus onset, but going all the way to 2 seconds post-stimulus onset to catch when people were speaking. So this was a picture naming task, and it takes usually under a second for people to name an image. So we were sure that we were catching the time that people were speaking at. And then we computed BSSCCA for each epoch separately, which is important because when we speak, when we produce different words, our EMG activity is not necessarily going to look the same, right? So you saw when I was saying tractor, it looked a certain way. When I'm saying house, it's going to look different, right? So that's why we computed BSSCCA separately for each epoch. And then we sorted the sources based on their degree of autocorrelation. And that we use a criterion to separate the EMG sources from the EEG sources as we classified them. So what we said is that the criterion we used was based on the fact that EMG has higher power at higher frequencies than EEG. And so sources were classified as EMG if the average power in the EMG band, which was approximated by being within 15 to 30 hertz, was at least 1/n, which of the average power in the EEG band, which we approximated as being between 0 and 15 hertz. So that's just the criterion we used in order to make that decision and say, okay, above this is EMG and under this is EEG, right? And we tried playing around with that criterion and to see how much it affected our results, and it didn't affect our results so much. Okay, so just to show you some results, so what it looks like on single trials. So this is a single trial of EEG at 5 different channels, And you can see, so the vertical dotted line here represents the vocal onset. And you can see the presence of a lot of EMG activity there, right when we're speaking, which makes sense. These are the BSSCCA components, or the waveforms of the BSSCCA components that we removed, or the time courses. And this is what a clean trial looked like. So it worked pretty well. Of course, I'm showing you a good example, right? Sometimes it didn't work as well, but I'm not going to show you the worst example. We also focused specifically on the CMG activity in other studies, which you can go and look at if you're interested. Now I'm going to show you a few slides on why it is that we think BSSCCA worked nicely for what we wanted to do. All right, so how do we know it works, right? Because that's always a big question. You don't want to be removing brain activity, right, when you're using these techniques. So it's important to be really careful about that. And so we tried to do that as much as possible. So this is the topography of the removed components, right? So when I showed you the time course earlier, that's the middle panel, right? So the middle panel here corresponds to the removed BSSCCA components. And this is the topography of these removed BSSCCA components. And you can see that there was a lot of activity here around the face, right? So, which makes sense because that's where the muscles are, right? We also looked at the average spectral content before and after BSSCCA and after 10 Hertz low-pass filter, which is pretty harsh, but is actually something that was used in the field. Maybe not so much today, but like a 15 Hertz low-pass filter is still something you'll see in the language production literature in EEG. And the problem with those low-pass filters is that they really cut down all of the activity that's above that, that cutoff, which is, what they're supposed to do. But you're missing, it's going to affect your EEG signal, right, in certain ways. And I'm going to show you one of these ways. So in red, you have the spectrum after the low-pass filter, and in green is after BSSCCA, and in black is the original, so the raw data, right? And so you can see here that BSSCCA is not removing all of the high frequencies, right, like a low-pass filter would do. So you still have that that presence of higher frequency activity. You're not seeing my mouse, okay? So you still have the presence of that high frequency activity here, right? It's not just completely removing it. But what is nice is that the spectrum regains its 1/f shape, right? That EEG signal typically has. And you get rid of that bump, right? That is the EMG activity. So we looked at ERPs. What did ERPs look like before and after BSSCCA and after the 10 Hertz low-pass filter? And what we could see is that the amplitude of these ERPs was better preserved after BSSCCA than after the 10 Hertz low-pass filter. So your 10 Hertz low-pass filter really cuts off the peaks of your ERPs, right? And we also compared our BSSCCA method to ICA. And what we used there, we chose to use this robust ICA technique. And we used it on each epoch separately with the same rationale as for BSSCCA because EMG looks different from one epoch to another. And we use the same criterion exactly to separate the sources that we call the EMG sources versus the EEG sources. And it didn't work that well, right? ICA was not, it actually kept a lot of the high frequency noise. That's the pink fuchsia line there, right? Okay, so we were pretty happy with BSSCCA, right? But of course, it's not a perfect technique, and sometimes it's gonna work better than others, and you're gonna be left with, trials with artifacts, and you're gonna have to reject them, so you lose trials. So recently, I've been working with people at SDSU to try to see if we could find a better technique. and think about, are there any other technique that can beat BSSCCA for the removal of speech-related EMG artifacts? So when I say we here, it's always Colin Farah, right, who actually did the work. He tried using this k-mean singular value decomposition algorithm, which is a type of dictionary learning algorithm. So it's machine learning, and I'm not going to be able to go into a lot of detail there. So if you're interested, you can email him, and he'll be happy to take your questions. But I'm going to show you a bit what he found. And it didn't work that well. So this is the first time that he tried to use it, pretty much. So he used it to remove the EMG artifacts. So basically it learns, right? It learns what the EMG artifacts look like. So by feeding it, this algorithm, by feeding it the actual EMG activity and seeing if it can find it in the signal and then remove it, right? But what happened is that it removed all of the low frequency activity too, right? So in red is after this k-mean singular value decomposition. So it was a little too harsh, right? So when he sent me that back, I said, well, we're losing a lot of the EEG activity, so go and try again. So it removed too much signal energy to be used as a general purpose algorithm. So then he went and he tried again. And what he did is that he actually first ran an ICA, and then he tried running this KSVD on the independent components, right? And then try to reconstruct the signal from there. And it was better then, it worked better, especially for blinks. So you can see here on this example, right, that we have a blink, and it's nicely taken care of by this KSVD on the ICs, right, on the independent components. For the EMG activity, it didn't really work. So he tried again, and Colin tried this time using a modified multi-scale method. And what he did is that he actually first did a wavelet or maximum overlapped wavelet transform to decompose the data using the largest number of scales possible. And then he used KSVD on those wavelets to find a sparse representation of individual scales. And then he retained the sum of these sparse representations corresponding to the data from 0 to 64 hertz instead of subtracting the sparse representation. So he kind of flipped it around, and instead of removing signal, instead of removing the sparse representations that he thought looked too much like EMG, he kept the ones that were between 0 and 60 hertz. And this time, it actually looked better. And so this is what you see here on, So this is what you see here on top, right? So in red is BSSCCA, and in blue is after his multi-scale method. And so now it's looking more like what we had. This is the raw signal, right? So there's definitely an improvement, and you can see a lot of this EMG. This is continuous EMG activity. It's not speech EMG activity, but it's still nicely removed. And so that's when he reached the end of his thesis. So we couldn't really then go and do a lot of tests to see, to compare carefully BSSCCA to this multi-scale method, but that's what we're going to do next. So this multi-scale method appears to be effective at removing EMG, but further study is needed in order to optimize the approach and see if it's better than BSSCCA. And you can email him for details, right? colin.farah@gmail.com, right? Okay, so moving on to EEG of single word production. Okay, so we did all of this so that we could use EEG to ask questions about cognitive processes, right? So that's where we were headed. So I'm first going to talk to you about this one study that we did during my PhD again, to just see if we could even look at ERPs during language production. And the answer is that yes, we can. So that was great. And this is what these are the ERPs that we found. And these are Laplacian transformed ERPs. I don't know if Boris is here or no. Okay. So, well, if you know Boris Bjorn, you'll do A. You know that you have to look at Laplacian transformed ERPs. Otherwise, you just go talk to him if you want to know why. But basically, those are what those are, right? So they're not your regular monopolar ERPs. They're Laplacian transformed, meaning that we're basically taking into account the activity or the signal at the neighboring electrodes to do a spatial derivation of the signal, which allows us to get activity that looks different from one electrode to another, right? So doesn't look all the same and homogeneous. It's actually giving you an estimation of the current source density. So we wanted to see if we could have nice Laplacian transformed ERPs when people were naming pictures. And so this is the study that we did. Of course, in order to really see if we could get good signal, we had people name 900, produce 900 trials. So it was very boring, and there were lots of repetitions, but people did it, and we got those ERPs. So just to show you, so this is these are the ERPs recorded at FCZ and CZ, right? Time lock to stimulus onset here, and time lock to vocal onset. And these are the ERPs recorded at FC5 here and FC6 here, time lock to stimulus onset and time lock to vocal onset. So we could see nice ERPs, and we did some analysis on them to see if they were actually bigger than noise, et cetera, and they are, so we were happy. And we also did source localization. So this is a source estimation. This is a surface minimum norm. We did time lock to stimulus presentation, where you see a lot of visual activity first, and then you see, as time progresses, this medial frontal activity that shows up. And time lock to vocal onset, you have this medial frontal activity that decreases as we get closer to vocal onset. And in parallel, you have this left frontal activity that gets stronger as we get closer to vocal onset, so which nicely matches the what you see in the ERPs. And we also did dipole modeling, so which worked nicely. Time lock to stimulus onset, not so nicely time lock to vocal onset. But so we could do a lot with this data, right? But it was because we had so many trials per participant, right? So since then, I have tried doing dipole modeling in the studies that I've done to try to ask more, like cognitive questions, and it's been harder. So when you have less trials, it's not as easy. So this is just to put some citations out there if you're interested in looking at studies that I did to actually ask some of these cognitive questions. So looking at speech monitoring in healthy speakers in patients with stroke-induced lesions, in deaf and hearing individuals during sign language production, and also looking at word retrieval in healthy monolinguals and bilingual speakers. So I've started working with bilingual or on bilingualism pretty recently, and I've learned from people who are experts in bilingualism that there are no true monolinguals, right? So that's why I have the quotes there. So, these are, of course, I think they're interesting because I did them, but I'm not going to talk about them a lot because my understanding was that this conference is more, about the methods. So I wanted to spend more time on the methods. And so I'm going to move on to talking about word sequence production. And this is a study that I did with Svetlana, who is here somewhere. Yay, there she is. And Bonnie Nazari and Bob Knight. So what happens when we say several words in a row, right? And it may seem like a very trivial question, and why am I even making a big deal out of it? But when you want to study ERPs that are time locked to each word, it becomes a challenge, right? You don't know how much producing several words in a row is going to influence ERPs time locked to each word. So this is what we wanted to ask. And we started with this tongue twister production study. And so what participants did is that they practiced saying these tongue twisters. Let's see if I can do it. Just rum, rug, see, just rum, rug, jump, right? So you have to practice saying it several times in a row. And you practice saying it with a metronome, and the metronome was beeping every 650 milliseconds. So that was to ensure that the words were spaced enough apart from one another so that we could look at what was happening time locked to each word, right? So you had the beep, and then you practiced, and then the beep stopped, and that's when we were actually interested in looking at the EEG signal. We didn't have the beeps anymore, and participants were still saying the tongue twisters, right? So they said them twice in a row. Okay, and then we looked at the EEG activity time locked to each word, okay? So what are we manipulating in this paradigm? The reason why we chose this paradigm is that it's very well studied in psycholinguistics, and there are effects there that we were expecting to find. And so here are the behavioral results. And what we were interested in here is seeing if we had our effects of cycle, right? So cycle corresponds to, each time you say the tongue twister. So the first time you say it, it's cycle 1. During the test phase, the second time you say it, it's cycle 2. And here is each word, right? So 1, 2, 3, 4, 1, 2, 3, 4 for cycle 2. And you can see that the error proportions increase as position increases and with cycle, right? Which makes sense because you're saying more and more of these tongue twisters, and the level of conflict increases as you progress through the tongue twisters. So we were happy to find these effects. We didn't have a clear effect of the phonological similarity manipulation, which I didn't really go over, but the tongue twisters were manipulated so that they were more or less phonologically similar. And of course, if they're more similar, the idea is that it's going to make it even harder, right? And we didn't really find that clear effect on behavior. But other studies have found it, so it's probably just a fluke here. Okay, so here are the grand average ERPs time locked to vocal onset. So this is taking all of the segments together for all of the words. And what we were interested in here is just seeing if we could see ERPs time locked to word onset, and we could. So we found this ERP here at FCZ and CZ, and we were interested in the error-related negativity, so that's why we looked at these recording sites. And we also found this left frontal activity that a reviewer made me give it a name, so now it's called a LLAN, which is a late left anterior negativity, right? Which maybe will change because that's a horrible name, but so this is the activity and this is the topography. So what we wanted to see is whether or not there was a return to baseline, right, between before the ERP and after. And this is what we found, right? For all of those, there was no significant difference between the amplitude here at the baseline and after the component. This was the case for these components and this one here. And in red, sorry, it's FC6, right? So just to show this is left lateralized. And the slopes were significantly different from 0, right, in the rising part of the ERPs. So, we were confident we had ERPs. But we didn't know whether or not these measures would change depending on the task conditions. So that's what we looked at next. And we looked at an effect of cycle. So remember, the first time you say the tongue twisters, cycle 1, second time is cycle 2. And what we found is at CZ, there was an effect of cycle, right, on the amplitude of this ERP, but there was also an effect of cycle on the return to baseline, right? So for cycle 2, the ERP there did not go back to baseline, right? Then we looked at the effect of position, so it's more noisy because we have less trials, of course. But we found here for FC5 there was an effect of position. So with increasing position here, you can see that the amplitude decreases. And there was also an effect of position on the return to baseline. So actually the ERPs did not go back to baseline. And then we looked at phonological similarity, and we found a small effect on FCZ, right? So again, on the amplitude, but also on the return to baseline, okay? And so the fact that we kept finding this effect on the return to baseline, made us wonder what was happening. And we looked at the whole sequence, right? And this is the average EEG signal along the whole sequence. With here is the time in seconds, and the vertical lines represent each word, right? And so what you can see is that, well, there's basically the EEG signal continues to rise across the sequence. It's not like it goes back each time after each word. It doesn't go back to baseline, right? So what does that mean for our findings on the ERP amplitudes? And so Svetlana ran some simulations to see what it could mean, right? And so these are theoretical ERPs, right, that return to baseline each time. And this is the average, right, of those 4 responses. And here are the same, but now with a slope that increases, right? And what you can see is that when you average those, and they have the exact same amplitude, all of these bumps, when you average them, now you can see that there is a difference, right? So the amplitude gets higher, right? And that is only due to the fact that the baseline rises, right? Is only due to that background continuous EEG activity that is added on to your transient EEG activity. And you can see the same thing if the slope decreases, right? So if the slope decreases, now your amplitude is smaller, right? Okay, so what does that mean, right? So we'll start with positive, right? It's possible to measure ERP's time log to each word in word sequence production, at least when words are spaced enough apart, which was also criticism of one of the reviewers. It's not like real life, right? We're saying, how often do you say tongue twisters 650 milliseconds apart, right? It's, yeah, it is. It's not like real life. But we first had to establish what it is that we could see when people were saying several words in a row. So we started like that. We found effects of experimental manipulations that could be observed on peak amplitudes. But the baseline matters, right? If the baseline shifts across the word sequence, then that's going to influence possibly the peak amplitude of your ERP's time log to word onset. So it could indicate something interesting, right? It could indicate that we have a mixture of sustained or slow rising EEG activity caused, for example, by interesting things like word sequence preparation or other cognitive processes that are engaged across the word sequence, and transient activity that maybe represent cognitive processes engaged at the single word level, but we still need a lot of studies in order to figure that out, right? So we're just starting to try to understand what happens when we are producing several words in a row. So the door is open to use ERPs to study word sequence production, but with a word of caution. Okay, I still have a little time. Yeah, 7 minutes, according to this. Okay, so intracranial EEG of speech production. And that's actually what Anne-Sophie wanted me to talk about, so I thought I can't not put it in there. So here you go. Anne-Sophie, you're welcome. So what does intracranial EEG tell us about word retrieval, right? So this is a picture by my colleague, Adine Flinker, that I keep reusing because I think it's so cool. So basically what you see here are the electrodes, the ECOG electrodes that are placed directly on the brain surface. So this is in patients that are suffering from intractable epilepsy. And so in whom it's not possible to know where the epileptogenic zones are. Maybe you already know all that, but just giving you some context. And so in these patients, neurosurgeons can either place electrodes that are perpendicular to the brain surface, and that's SEG, or they place electrodes that are on top of the brain using grids of electrodes, and that's ECOG, right? So this is a case of an ECOG patient. And so the advantage of intracranial EEG is that you get both spatial and temporal resolution, right? So yay, you get the best of both worlds. And so we were trying to see if we could, get some answers about which brain regions are important for different aspects of speech production. And I did this when I was doing my postdoc at UC Berkeley. And I was fortunate enough to get data on, how many is that? 7, I think, patients with ECOG recordings. So we had 5 with electrodes on 5 or 6 on the left, right, hemisphere, and then we had 2 in the right with coverage in the right hemisphere. So each color represents a different patient. And we had people produce this task, which is a block cyclic picture naming paradigm. And basically what people do is they just name the pictures that are presented one after another. Sometimes these pictures are in semantically homogeneous blocks, like here, they're all items of furniture. And sometimes they're in semantically heterogeneous blocks, so they're in they're from different categories. And the expected effect is that people will be slower and they'll make more errors in the homogeneous blocks than in the heterogeneous blocks. And that's a particularity of speech production. If you were doing something related to speech perception in the same type of paradigm, you would get the opposite effect, right? So the idea is that in speech production, you have to choose, right, among different possible words. And those semantically related words are going to interfere with one another when you're trying to select one. And so that's why you get what we call the semantic interference effect. I'm brushing really fast on all the cognitive aspects because this is a methods conference, so I want to get to the methods. Okay, so here are the behavioral results. And so I didn't say this, but people name the pictures several times in a row. In our case, they named them 4 times. And what we typically find is that this difference between homogeneous and heterogeneous blocks increases with presentation number, right? At least it's not there in the first presentation, and then you see it later on. So this is what we found. And the mean reaction times were longer than what we normally see or what we see in, people that are your young controls, your college students that are coming to the lab to participate in the experiment. They're going to be about, 600 milliseconds. It's going to take them 600 milliseconds to name a picture. But, these are patients in the hospital setting, so it takes them a little longer. So we focused on the high-frequency broadband activity, formerly called high gamma, which is why you have the HG there. We now call it the high-frequency broadband activity because it's not actually an oscillation. We think it's actually more of a broadband signal, as you can see here on the left. Okay, and we focused on the section between 70 and 150 hertz. The advantage of this activity is that it's pretty clearly visible at the single trial level, which is what you see here on the right. It's fairly focal, so it doesn't spread as much as the lower frequency activity, and it's been associated with a lot of cognitive processes. And so this is just to show you why people like it so much. So this is going to show you the high-frequency broadband activity as we're naming the picture. So at minus 500 milliseconds, you're going to see the fixation cross, and you're going to see some preparatory activity in the frontal cortex. This picture is going to come on now, and now we have activity that starts appearing at all these different sites. And as we get closer to vocal onset, which is going to be soon, you're going to see more and more activity in the posterior STG and in the motor cortex. Okay, so that's why people like it so much, because it's so precise, right? You can see it; it's really localized, and it's of course it has a great temporal resolution. So that's why we wanted to look at it. And so this is a snapshot of the results. This is time locked to the stimulus, and what you see here. So at each dot here is an electrode, and the different colors are for whether or not the effect of semantic context was in the direction of semantic interference or in the direction of what we call semantic priming, right? So if the dots are red, it means that at that electrode, the semantic context effect showed that there was more activity in homogeneous blocks than heterogeneous blocks. And for blue dots, it's the opposite, right? So there was less activity in the homogeneous blocks than in the heterogeneous blocks. And this is just for visualization. We didn't do analysis like this. This is just to show you, what it looked like. And so what you can see is that there are red dots and blue dots pretty much all over the place, right? So it's hard to make sense of it. And that's often the case when you look at intracranial EEG. It's not clear cut. So basically having the best of both worlds also gives you a lot of data to work with, right? And that can be a challenge, and it was certainly for us. This is time locked to the stimulus. This is time locked to the response. So same thing, showing you the evolution of these effects across time. And so the way that we actually analyzed the data, that we did statistical analysis, is that we computed one model, one general, one linear mixed effect model, time locked to the stimulus and time locked to the response in the frontal cortex and in the temporal cortex, right? So we had basically 4 models, and that was it, right? But the cost of doing that is that we had to lose some spatial information. And so that's kind of our choice that we made, because we had to make a choice for statistical analysis. There are different choices that can be made at that point. For the temporal lobe, we divided the temporal lobe in these 4 areas. And for the frontal lobe, this is the choice that we made, right? And basically the models that we ran, I'm going to show it to you. It's at the bottom there. So this is the model that the type of model we ran, right? So we looked at the effect of semantic context and its interaction with repetition and brain structure and time window. And we also checked for an effect of laterality. So the pictures were, the images were presented on the left and the right of the fixation cross. And we controlled for the picture that they saw, and we also controlled for these effects within patient. And so what we found with these big overall analysis is that in the temporal lobe stimulus locked, there was a semantic interference effect that increased with time overall, but it was in fact confined to the ITG. And then in the frontal lobe, where the most interesting was actually time locked to the response. So yay, I'm so glad we did all this work in scalp EEG to figure out how to look at ERPs time locked to the response, because that's really where things seem to happen in the frontal cortex. We found a main effect of semantic context, and semantic interference decreased with time, and it was maximal before 350 milliseconds pre-vocal onset. So those are the results, the main results that we found in this study. And I'm almost wrapping up. So the cognitive interpretations that we had were that the left inferior temporal cortex seemed to be involved in lexical access as indexed by semantic priming, and then selection as indexed by semantic interference. So maybe it's actually involved in both aspects of word retrieval. The left frontal semantic interference effect was more widespread and sustained. Maybe that indicates a role in cognitive control of lexical selection. We can't really say, but we could with additional studies that we did on patients with lesions in the left frontal cortex, and there we found that indeed they seem to be impaired in the cognitive control aspect of lexical selection. And another question is, the semantic interference effect is more widespread than predicted. Does that mean that lexical selection is both internal and external to the language system? And we're conducting studies to try to understand that. But what about the methods, right? So that's what we're here for, right? At cutting edge EEG, right? So using IEG data to study cognitive processes suffers from having to choose between spatial and temporal resolution, even though we have both, right? So it's really sad because we have to choose between one or the other, even though this is the technique that gives us both. unless we choose the brain regions that we're interested in, right? So unless we do an ROI type of analysis, but then we risk missing relevant activities in other brain regions. So it's again a choice. And I'm looking forward to hearing other potential cutting edge solutions. So thank you for your attention. Thank you to my mentors and to the students and to all of you and the participants, of course, and the sources of funding.
Thank you. We are running late, so we have time for very urgent questions. Otherwise, we can have them at the discussion session at the end of the symposium.
Hi, thank you for your talk. I was wondering regarding cleaning the data from speech artifacts. You focused mostly on high-frequency activity, but have you also checked and what do you see kind of in the 2 to 15 hertz range, which is also pretty characteristic of speech?
You mean for the IEG study?
No, in terms of the EEG study, so cleaning the data.
Oh, yes. I mean, we focus on the lower frequency bands naturally by looking at ERPs because they're actually issued from lower frequencies. So we didn't analyze the higher frequencies, right? We only looked, that was only to estimate which were our EMG sources versus our EEG sources. But you're right, EMG activity impacts lower frequencies too. They would be removed as well with this technique, right? It's just our criterion in order to select the sources, if that makes sense. I'm happy to talk about it more. Yeah. There's one over there. Yeah. I just saw her hand raised up before yours. That's why.
That's okay. Thank you very much for a very nice talk, very complete, lots of data to think about. Tying it back to the very beginning of your talk, when you're discussing your method of cleaning, this is for, is it strictly related to experiments that are looking at speech production? And I'm asking that question because you gave the example that you're actually going to look at the different articulatory movements that are behind each different item that you're producing and that you're putting that into your model. And I'm wondering if you're looking at just passive listening to speech, you're going to need that level of detail.
For sure, yeah. If you're looking at speech perception, you're not going to suffer from the same, artifacts. So you wouldn't have that problem. Yeah. Yeah, but you will have sustained EMG, from frowning, from what you usually get in EEG, right? And BSSCCA can take care of that, so it can save you some data. Yeah.
Have you studied any causal effects between the encoding areas and the reception areas?
No.
Like connectivity between those areas?
Oh, connectivity. So I've tried to do some connectivity, analysis, simple correlations between the signal recorded in the temporal cortex and in the frontal cortex with the IEG signal. And then we tried some more sophisticated graph signal processing with people at SDSU, and I didn't present that, but I'm happy to talk to you more about it if you're interested, to send you the paper. Yeah.
Thank you. We are going to stop now. So we have the coffee break and we will start again at 11:30.